In the first video, I shared the big picture, why patients and pharma are stuck and why we need a new bridge between them. Now, let's go deeper. Let's talk about how this kind of patient drug screening can actually transform both oncology and drug discovery. Let's start with pharma. Every year, pharma companies synthesize and screen millions of molecules. The goal is to find the few that survive the brutal drug development pipeline. But here is the problem. Almost all those screens happen in the engineered models like CRISPR-edited immortalized cell lines, organoids, and similar systems. They don't fully reflect real tumors, and animal models don't capture true human biology. By the time a drug reaches clinical trials, we discover the truth, and 9 times out of 10 it fails. Now imagine a different path. Imagine if pharma could test molecule libraries directly on patient cells, early, rapidly, at scale. Instead of spending years on molecules that will eventually fail, companies could triage early. They could see with actual patient biology which molecules have the promise and which don't. The impact: billions saved in failed trials, higher success rates, and perhaps more importantly, a data set of unprecedented value. Because here is the kicker: every molecule patient test creates data. Multiply that across millions of molecules and thousands of patients. And what do you get? The most powerful data set oncology and pharma have ever seen. This data set doesn't just help with discovery, it trains AI to actually predict real patient outcomes, not guesses based on mutations, no lookup tables, not patterns from animals, but predictions grounded in patient biology. That is a kind of AI that can transform drug development. Now let's flip back to patient side. Genomics has given us incredible insights into mutations. But as we know, the same mutation doesn't always mean the same drug response. That's why patients often go through multiple lines of therapy before finding one that works. And by then, the time has been lost. With patient cell screening, doctors can test multiple therapies on patient samples in parallel and see directly which ones help the cells recover. No trial and error, no waiting for months to find out. Just evidence-based decisions made faster. There is another huge angle, which is repurposing. Pharma shelves are filled with molecules that didn't make it to one indication. But what if some of those same compounds work beautifully in unexpected patient subsets? Patient cell data makes that visible. It breathes new life into shelved molecules. It accelerates repurposing, not by chance, but by evidence. Here is the thing: none of this happens in isolation. It's not about one company or one lab, it's about an ecosystem. Pharma, clinics, labs, data scientists, all working together. Because only when we connect all these dots does the real impact emerge. So, here is where we are. We know the problem: trial and error medicines and failing pharma pipelines. We know the missing link: large scale patient cell screening that serves both sides. And here is my message: this isn't a dream. The technology exists, the collaborations are beginning, but biggest breakthroughs will come from those who join early. If you are in pharma, biotech, oncology, or advanced data science, I invite you to connect. Let's explore how we can build the future of precision oncology and drug discovery together. The future isn't far away, it's already here. The question is, who will help scale it?